Okay, so I won't have any trouble convincing you that I'm studying both males and females, and you'll see that in spades. I want to talk today about an animal we've been working on for some time because it has really unusual properties that we've been exploiting to really get a handle on how the behavior changes the brain. So to do this, you need an animal that you can look at many animals simultaneously, and they focus primarily on sex and food. This also explains why we have a lot of undergraduates who want to work in the lab, because that's sort of the name of their lives. So, and we want to also look at many animals simultaneously, and this is really an important feature. You'll see that doing this, we're able to actually get much more mileage from the animals. And we look at this fish, which is called an African cichlid fish. Here are two males fighting. And I'll do a couple of things today rather quickly, but first I'll tell you a little about the natural social life, and then something about how social status changes the reproductive life of these animals. Finally, what do they actually know? Because many of you may be suspicious that if you're studying fish, they may be like your goldfish who can barely tell up from down. These are not that kind of fish. These are fish who are really very smart. And then how does social information get into the brain? How is social information transduced and causes cellular molecular changes, and how fast can this happen? Which I think is a model for actually all organisms. And then finally, could there be epigenetic regulation of social status? This will be, I think, interesting to you to see that there is some evidence now that epigenetics may play a role in social status. So here are the animals. Here's a dominant territorial male, and here's an animal that may be a female, but it may also be a non-dominant male. So these animals can switch back and forth between these two states, and they do that regularly, and that's sort of the name of the game here. So they live in Africa, in Lake Tanganyika, one of the great rift lakes here. This is a really unusual setup. These rift lakes lie along this tectal rift. You can see that Madagascar broke off, and those of you who become interested in this fish will have to hurry, because this is going to break off and become another continent, another 93 million years, and then we're in trouble. And previously they'd been studied by Max Pohl, shown with his major research tool, the depth charge. So he blew up large volumes of Lake Tanganyika when the Europeans were exploiting this whole area of Africa, and shipped back to Belgium all the fish he found. There wasn't a lot of behavioral information in his work, because it was mostly pictures of fish with their fins blown off. So basically, I went to Africa to study them and discovered, and I'm here today because they live in shore pools along the edge of the lake. And they live in these pools, which I've marked here with stakes, and these are territories where an individual male will actually defend a territory and try to lure into that territory a female to spawn with him. And they have to spend on this female food, and they are a unique group because only about 10% of these males actually have territories. The other 90% of the males actually have to pretend they're females. They will go in, they'll hang out with females out here at a school. They'll go in and pretend they're females, go into the spawning area, eat vigorously, and then the male says, let's go into this pit. And the male faking being female will say, you know, I really have other plans, and speed away and then do this somewhere else. The females, of course, in this system, like in all systems we know about, are completely in charge. And they can actually swim into this territorial area, choose a male who's large, vigorous, moves quickly, and can provide them a lot of food, and then they will choose to spawn with them. And territorial males basically all day do the following things. So they're pretty active. Here's a male who's doing okay until a challenger comes up, and he'll actually attack this guy. This is the challenger here. And you'll see he's taking over a part of this male's territory, and he'll now take this piece of the territory, and this guy will be subjected to a much smaller territory as this guy turns on his eye bar and takes over this spot. You'll have noticed, too, that there are lots of animals watching him, and that turns out to be part of what they do. They watch each other a lot to figure out what to do next. Spawning is interesting because the female lays the eggs and then picks them up in her mouth, producing a problem for fertilization, which has been neatly solved by these anal fin spots. And the male will actually swim up in front of the female, put the fin spots down in front of her, put the sperm on top of them. She'll collect those fake eggs and fertilize the eggs in her mouth. This is what it looks like here. So she's now laying those eggs and picking them up quickly. And he will, this was made by National Geographic, so he wouldn't do this normally. He swims off and displays his prominence to the viewers. But normally he would go straight into presenting his anal fin spots, and then she will pick up the egg dummies and pick up the sperm and fertilize the eggs in her mouth. So she then carries them for 2 weeks, and that gives us a big supply of animals, basically. And it's really a wonderful system, which is regulated by the females indirectly, but by the males fighting over the territories. So there are these two phenotypes that are quite different. This dominant or territorial male who's reproductively active and the non-dominant male who's not. And we've mapped many aspects of their difference, but the most important thing is this is entirely under social control. So whether you're dominant or not depends on your environment, who is bullying you and who's actually in charge. And we can show that there are differences in phenotype, difference in a class of cells called GnRH cells that regulate reproduction in all vertebrates, and in the receptors expressed and the gonads. And basically, we can convert these animals back and forth in the lab between dominant and non-dominant. We do that just by putting them in the presence of larger conspecifics, and a dominant male will rather quickly then become non-dominant and lose his coloration and do various other things that you'll see in a minute. So when we do that, and we look at the size of these GnRH cells, which is really a proxy for GnRH, we can show that when they're non-dominant, they actually have small GnRH cells, and when they're dominant, these cells grow in size. And when they lose that dominance, they will shrink those cells, and when they gain it, they will get those cells back again. So in fact, there is this massive change in the brain as a function of their social status, and it's driving the reproductive system. And it also changes another thing, which is the actual extent of their dendrites. So the dendrites of the GnRH cells in a non-dominant male look like this, and here they look like that. And they're quite different in their functional attributes as well. So when you're a non-dominant male, these cells are disconnected, and when you're dominant, they seem to be connected. So if you look at that in a non-dominant dominant male, and you put a cross-correlogram, which measures the amount of firing of these cells, these are firing in synchrony, whereas these are not. And that's a prerequisite for actually being able to reproduce. So all of these are driven by this social behavior. So the question now is, do these animals know, what do they know, and how do they use this information in order to govern their lives? So I'll show you just a couple of examples. They have a kind of behavior that you probably all are aware of, which is called a contingent behavior. So here's a dominant male. This is when he's in his shelter. And he comes out, and when he's out, he attacks other animals. So he's basically being a bully pretty much all the time when he's not in his shelter. And this is witnessed by other animals. Here's an intermediate male who, notice, will not come out when this guy when he's in his shelter, he will actually attack. And so he'll take advantage of the absence of this guy visually in order to make moves on females and also to try to express himself in a more dominant form. So this is a kind of a contingent behavior seen previously in primates, in faculty meetings, in a lot of playgrounds, places like that. It has a lot of similarities across species. They can also infer social rank just by observation. So a really wonderful experiment done by an undergraduate showed animals as staged fights and proved that they could actually, when they were trained that A would beat B and B would beat C and C would beat D and D would beat E, he then asked them, well, would B beat D, a fight they had never seen? And they could actually do this. So they had a transitive inference, which is a skill that you all acquired at age 3, and they can actually do it. And they use this to spare themselves from having to fight everybody by knowing who's going to beat whom. Okay, so the other thing that they actually do is something that's quite interesting. The females, the short answer here is that females never like to choose losers. So females are watching a male. Here's a female with a large belly. This is a staged photograph in which she's watching these two males fight. And we asked the question, what would happen if she saw them fight? So here they are choosing between two males. Here's the female. And she will choose one of them. And then we will actually stage a fight where the guy she chose either wins or loses that fight. And she's basically watching the fight and getting information about the chosen male. She's not actually doing anything with him. She hasn't been able to spawn with him or get close to him. She's behind little pieces of plexiglass. But then we will test her to ask what happens to her brain when she's seen a male that she has chosen either win or lose a fight. And we look in areas in the brain that are common across all vertebrates using real-time PCR, which is a measure of expression of genes, immediate early genes that are turned on when they're activated. So we do this by looking at the social brain. It's a set of brain nuclei. I won't go through this in detail, but all the pieces that are present in mammals are also present in fish. That's the beauty of evolution. So we're looking at the same nuclei that would probably play a role in any female choice about males. And so when we do this and we actually look at the outcome at the social behavior network with this collection of nuclei that are responsible, we'll find a real interesting signature. So when she sees her preferred male win, a number of these nuclei will turn on. And she's turning on her entire reproductive axis upon seeing the male she chose win a fight. And when he loses this fight, she'll actually turn on the lateral septum of a nucleus which is common across all vertebrates that is actually associated with anxiety and processing anxiety. So seeing your preferred male win has one outcome, and seeing him lose has another outcome in the brain signatures. And we then tested these animals again, and the females, as you might guess, being a largely female audience, never make the same mistake again, and they will never choose the loser, even though they've seen him a couple of times. And of course, in males, a comparable kind of work that we've done is, I'll just show you one example. The male aggression really depends on who's watching. And this is not unexpected, but if a gravid female is watching a male, he will actually be much more active and fight more vigorously than when, for example, a larger male is watching him, where he will reduce his fighting ability. So they're actually attending to what's going on in their social system and making a difference count. So we then asked, well, how is this transduced into the brain itself? So we knew that the social environment, the perceptual environment, led to a change in phenotype, which in turn led to this change in reproductive capacity. So we did something which I refer to as a bullyectomy, where we put animals into a situation and we then remove the dominant male and watch what he does in response. So I'll show you what this looks like. Here's a social group. You have to keep these animals socially attentive. So they're in two social groups, and you'll watch them here. And then the lights go off, and the next day at sunrise, he wakes up, and the guy is not there. So here's what it looks like. Sort of your dream as a child, having the bully from your neighborhood extracted from your neighborhood. Never occurred for me, but it works for these guys. Here's the guy who's the non-dominant male. Here's the dominant male who's got a shelter back here. And he will actually chase and try to court these females, and basically full-time bullying activity. And he'll do this all the time. He'll sometimes fight with neighbors who are through the glass here, or just chase a female who's not interested in him. And they basically spend their time looking out the window, wishing he weren't there. And ultimately, they will all be in response to his bullying. The male will be suppressed. So now, if you imagine the moment when this non-dominant male wakes up and the bully is gone, this is him acting when the dominant male is no longer present. And sure enough, he will begin to attend to this shelter. We know personally he's been suppressed long enough. He probably does not have mature sperm, but he will pretend to be a dominant male, and he will turn on his eye-bar and chase these females around and pretend to spawn with them. So he's acting it out, even though he probably can't do as much as he could were he a real dominant male. So we then wondered, of course, how quickly does this information get into the brain? And you can, in contrast to working with humans, for example, actually look at the brain directly and ask what happened. So we looked at these GnRH cells, and basically we used a map of the immediate early gene EGR1 in the GnRH-containing cells. So these are the cells that are changing size in response to the social advance. And what we discovered was that after 20 minutes, he actually was turning on gene expression of the GnRH gene, EGR1 in the GnRH gene cells. And EGR1 is a transcription factor for GnRH1. And we now know that he's able to respond to the social circumstance and change gene expression in a matter of minutes after recognizing this social opportunity. So in fact, they're responding really quickly to this perception of change in their social lives. And all of it's driven by their awareness of their social status and how they play this social status out in respect to other animals. This just shows you the data that goes with that. So let me now just turn to one last quick thing. We began to wonder whether there might be the kind of change in methylation, which is a kind of gene expression control that you just heard about in these animals. So we looked at two males who had never been dominant previously, and we had three conditions. They either received a control injection, and there, as you might imagine, one became dominant and one didn't. It's basically sized match animals who would ultimately either win or lose depending on chance. We then took some animals and injected them with methionine, which is a methyl donor, and part of the methylation process And sure enough, in those cases, it biased animals towards becoming dominant. And then we took animals and injected zebularine, which is a blocker of this DNA methylation process. And there was a change where they became biased to becoming non-dominant. So it could well be that there is a methylation process that is a change in gene expression that's occurring as a function of these social interactions. And we're now trying to do whole genome sequencing to look at whether or not this is the case. So all I wanted to say as well is this was not, we could not explain this based on the behavior. The behaviors were not different between a normal animal becoming dominant and an animal that was injected with methionine. Similarly, animals that became non-dominant were not different from those who did not receive the drug. So we think that this is happening, and we think also that it's really important to remember that when you do this, you can actually look at the genes themselves. Here's the GnRH gene. I mentioned before that EGR1 is a transcription factor, and we discovered that it was actually increased, its methylation was increased in response to zebularine injection. So it may be that the control, we'll be able to track the control all the way down to the genomic level and see what's happening during these social encounters. So we're now looking at what gene networks are assessed in during social ascent. We're taking out the cells, the GnRH cells, and looking at their gene expression patterns. And Scott and Caroline are building a transgenic ciclid, which will allow us to do things in an even better way. And we're thinking now that the behavior changes the brain in a really important way. There's a social life that's really changes, I didn't talk about this, but cells, receptors, and circuits, and genes in ways that we can now measure directly. And probably more importantly, animals collect information on each other. And this is now turning out to be true in many different species. So it's not just that in this fish, animals are watching what one another do, but all social systems require information, and information is important, and information is something that you use to control your behavior, and these animals are doing exactly the same thing. So social information can get into the system and change gene expression. The example I gave you with the female, that was only information she was using, and it was changing gene expression in her brain. So I think we want to say that aggressive dominant males have different brain circuitry than non-dominant males, and we'll be anxious to use some of the probes that Neuroscience is generating to take a look at those in our fish, because they're likely to be conserved. And I'd like to end by thanking my wonderful lab group and people who paid for this work, and for your attention.